I am in the Mosher lab, and very broadly, what we study is the plant-specific RNA-directed DNA methylation pathway, or RdDM, using the model organisms Arabidopsis thaliana, or thale cress, and Brassica rapa, which is field mustard. And this pathway is responsible for de novo DNA methylation of transposable elements. So what exactly does RNA-directed DNA methylation do? So I'm just going to walk through the key players in the pathway that are relevant to my project. First, we have Pol4, which is an RNA polymerase, which transcribes a single-stranded RNA. And then this strand is made double-stranded by another RNA polymerase called RdR2. And so you get this long double-stranded RNA, which is shortened or diced by a protein called dicer-like 3, which results in these small RNA duplexes, which consist of one strand that is 24 nucleotides long and another strand that is 23 nucleotides long. And then these duplexes are loaded into an Argonaute protein called AGO4, which then goes on to interact with more molecular machinery, which is actually quite a few proteins, but I've kind of generalized here into this one gray shape for conciseness, which then methylates DNA. And so that's kind of the general process. And so my project is based off of a previously published observation that in vitro, RdR2 will actually leave an extra nucleotide at the 3' end of its transcription product. And this kind of has some serious implications because most small RNA mapping analyses require an exact match between the small RNAs being sequenced and the reference genome. So you can imagine if all of your 23 small RNAs have this extra nucleotide on them that doesn't match the genome, then these would all be thrown out as junk during the mapping stage, even though they are, in reality, real small RNAs that were transcribed. So that would have some serious effects on downstream analysis. So my project is basically using exploratory data analysis to see if we can identify these RdR2 dependent 23 nucleotide reads in actual sampling data, as opposed to just in vitro in a test tube as was originally shown. And so for brevity, we call these RdR2 dependent 23 nucleotide reads 23 stars for conciseness. So this is basically a pipeline for how I isolate 23 star reads from small RNA data. So I start with raw small RNA sequencing data files, which are originally ligated to adapters for sequencing. So I use a command line tool to remove these adapters. And from there, I can use a Python script to select 23 nucleotide reads, and then use another command line tool to filter out a variety of different contaminating RNA reads. And then, so 23 star reads are thought to have this untemplated nucleotide. So what I want to do is get all of the reads that are 23 nucleotides long that do not map to the reference genome using a command line tool. And then from there, I can trim these reads by one nucleotide from the 3' end. And from there, I can align these reads to the reference genome using a small RNA specific alignment tool. And this gives me a set of reads that I can then compare to other data sets in R or visualize in a genome browser or otherwise analyze for different characteristics. Now I'll briefly cover some of my findings, which are not super exciting because basically we found that really we didn't seem to see much evidence for these 23 stars. You can see that 23 stars make up a very small portion of total small RNAs. So you can see that regular, just normal, non-trimmed 23 nucleotide reads are much more abundant than these supposed 23 stars. And I also looked at RdR2 mutant small RNA sequencing data and found that 23 stars were not especially depleted compared to different size classes in both leaf and ovule tissue, which sort of contradicts the idea that 23 stars are produced by RdR2. However, I also found that there's a small subset of loci where 23 stars are produced at significantly higher levels than would be expected relative to regular 23 nucleotide and 24 nucleotide reads. So we're kind of concluding that 23 stars are either not produced by RdR2 as seen in vitro, and that this may have just been the result of the experiment being done in vitro rather than in vivo in the first place, or perhaps 23 stars are RdR2 dependent and just degrade incredibly quickly relative to their 24 nucleotide counterparts. And so we're currently looking into possible ways to try and determine one or the other. And so finally, why does this matter? My project specifically is hoping to contribute towards a basic research understanding of RdDM, and RdDM itself is a beneficial process to learn about as it is implicated in many different cellular processes, which are sort of summarized here, including as a pathogen defense mechanism. And further, it's been sort of proposed that RdDM could be used in a potential genetic engineering method for crop development. So knowing sort of a more fine-grained understanding of the exact system is very helpful for those kinds of applications. And these are my references. Thank you.